Let me go to today's speaker, Dr. Ronald Davis from the Scripps Research Institute, whose topic is Alzheimer's disease, causes and potential therapies. I have a very long resume for Dr. Davis, but I'm going to give you a short resume for him so he has more time for his talk. Dr. Davis is a professor in the Department of Neuroscience at the Scripps Research Institute here in Florida. He's also adjunct professor in the Department of Psychiatry at the Baylor College of Medicine in Houston. He's a leading expert on the biology of memory formation and the brain disorders that disrupt memory. His current biomedical research focuses on the biology of memory formation, mood disorders, and the development of therapeutics for Alzheimer's disease and related disorders. He was recruited as the founding chair for the Department of Neuroscience at Scripps in 2009 and grew the department from 5 neuroscientists to more than 100 studying brain function and dysfunction. He stepped away from that chair position in early 2018 to focus on his research program. Ladies and gentlemen, Dr. Ronald Davis.
First, I want to, of course, thank Bob and the whole group of the Science and Technology Club for inviting me to come and speak to you. It's, I'm a science nerd, I think, like a lot of the members of the Science and Technology Club, which in my case means that I really think pretty much 24/7 about the brain and how it works and what goes wrong. The human brain, as well as other brains of other animals. And I'm sure many of the members of the Science and Technology Club are also nerds, given they are members. But that's okay, because being a nerd is currently fashionable. It didn't used to be fashionable, but it is currently. In fact, my son, who lives in Houston, he's now a partner at Deloitte. When he introduces me to his friends, he doesn't say, John, this is my dad, he's a professor, or John, this is my dad, he's a scientist. He says, John, this is my dad, he's a professional nerd. So that's his introduction to me. But thank you very much for coming. I know it's a lousy day, and so many of you basically waded through puddles and so forth to get here, so I appreciate you coming. The other thing I have to commend you on what I understand about Palm Isles, not only the Science and Technology Club, but how vigorous this community is. I'm constantly asked, and this always comes up, usually after a presentation, but sometimes during, what can we do to keep our brains healthy? To counteract the effects of aging and Alzheimer's disease and Parkinson's disease and essential tremor and all those other diseases that affect the brain. And the bottom line is right now, the best thing is both exercise, physical exercise, and mental exercise. So one of the things we'll just briefly mention is social engagement is protective against Alzheimer's disease and mental disorders. So what you're doing here, and I understand it's very, very hard to find a time slot for this theater because of the amount of social activities you have, is terrific. It's really the best thing you could do to help keep your brain healthy as we age. And as of course, modern medicine has been terrific in extending our lifespan. But what hasn't happened is modern medicine hasn't kept up with brain health. And so that's one of the reasons you see a high prevalence of brain disorders currently in our society is we are living longer, but brain health hasn't kept up yet. And of course, that's one of my jobs is to try to make sure that we do catch up with modern medicine. So just Bob talked a little bit about my history, but I just want to give you a little bit more so you know who I am and where I came from and why I'm here in Florida. So prior to coming to Scripps, Florida in 2009, I was recruited, as Bob mentioned, as the founding chair of the department. And my job there was to really recruit and build the neuroscience department. And we did this over a period of really 5 or 6 years. And it's a very vigorous department right now with more than 100 neuroscientists studying how the brain works and what goes wrong in brain dysfunction, brain disorders. But before that, I was a professor at Baylor College of Medicine for about 20 years. And so there I had a number of appointments. And as you see here, I had an endowed chair, the Welch Professor of Science, in a variety of different departments: neuroscience, and molecular and cellular biology, and psychiatry, and genetics as well, because my research covers very broadly all those aspects. I was also the director of a center for memory and learning at Baylor for about a 5 or 6 year window, which was we were basically pushing forward the development of a center to really understand how memories are formed in the brain and how they're learned and how forgotten. And then also I was the vice chair of research in the Department of Psychiatry for a number of years, promoting psychiatric research and brain health. And so those were the platforms that I had for both research as well as administrative activities in order to build the research infrastructure towards modern medicine and helping to bring therapies for brain disorders. Prior to that, I spent some time at Cold Spring Harbor Laboratory, and I think this is important. It is ingrained in me. So I was recruited at Cold Spring Harbor Laboratory as a senior scientist. This is a laboratory on Long Island. At that point in time, it was headed by Nobel laureate Jim Watson. Watson and Crick fame, DNA double helix. And so Jim recruited me, and one of the things he taught me very early, and taught all of us, was that it's our obligation as scientists to reach out to the public and tell the public what we're doing and why we're doing it. For after all, most of our research is supported by grant applications we submit to the federal government, to the National Institutes of Health, and they essentially pay for everything that goes on in my lab right now. So I am obligated to you, and I appreciate you coming, because it gives me a chance to tell you what we're doing in my lab and why we're doing it. And that was something that Watson ingrained upon all the faculty members here, and so for that reason, I do give quite a few public talks for that reason, because I think it's important, it's our obligation to do so. So let me now turn towards our two areas of my lab. One area that I won't focus on is really we work on how learning occurs. What happens in the brain when an animal learns information? That means what happens not only within the circuitry of all the neurons that we and other animals have, how they communicate with one another, but also down to individual genes that influence our ability to learn and remember, the molecules that basically you find in neurons that promote learning and memory, and not only learning and memory, but also forgetting. And we've discovered in recent years that the brain is designed to forget, which makes sense when you think about all the information we learn during the day, that it makes sense that we just chronically remove information unless something comes in and says, this piece of information is important, remember this. And for that general research, which is basic neuroscience, we employ a small insect called Drosophila melanogaster, or fruit fly. Many of you have seen those. If you leave a banana peel out in your kitchen counter, you're going to collect a few by this afternoon or by tomorrow. But it's absolutely amazing how much we have learned about how brains work by studying these model systems like fruit flies, or even some labs study worms called C. elegans. We also study mice and rats, of course, but flies are really a wonderful entree into understanding the genetics, the genes that are involved in building the brain and function of the brain to allow animals to learn information, to remember that information, and to forget that information. And there is between this half of my lab, which studies the basic neuroscience, and the next half, which is really working on therapeutics, you're not going to be able to see very much on the slides. My apologies, there's a lot of light for the video presentation, but what you see here is a neuron growing in culture. It's a mouse neuron, and just by background, this part right here is the cell body, often known as the soma, this yellow part, and this is the command center. This is where the nucleus is held. This is where the DNA for each cell is held as well. And what you see, and you'll see more of this, is really what you can't see very well because of the lighting, is there's a whole bunch of processes, extensions, that move like an octopus arms. And these are the axons and the dendrites that each neuron extends in order to have a synapse, which you recall, synapses are the specialization on axons and dendrites that allow neurons to communicate with one another, to talk to one another. So we'll talk more about that in just a minute. But this is the second half of my lab. Half of my lab works on the basic neuroscience, of learning and memory, half works on developing therapeutics for brain disorders, brain dysfunction. And that's what we're going to focus on today, is really Alzheimer's disease, what we know about Alzheimer's disease, its prevalence, what we know about its causes, and what the scientific community has done in the past in terms of developing therapeutics, and the tack that my lab is taking towards developing new therapeutics for not only Alzheimer's, but all neurodegenerative disorders, including ALS, Parkinson's disease, and so forth. And we'll come back to that in just a few minutes. But first, let's talk about the problem of the brain, memory in the brain. So your brain, each of our brains weighs about 3 pounds in our head. And if you haven't thought about it, you should think just briefly about what your brain does for you, because everything, all the information you are processing currently, all the visual information, all the sound, the auditory information, everything you're feeling right now, whether you're happy or sad, whether you're elated, your perspective plans, what you're going to do this afternoon, what happened a week ago, what happened in the past, your memories a decade ago, 3 decades ago. This is all due to the brain. So in essence, the brain is really everything we are. In fact, I think you could put it in the phrase that everything else about us, our arms and legs, these are all accessories. The brain is what makes each one of us an individual, and basically encompasses all of our thoughts and all of our memories and everything we know, we think about ourselves and how we perceive the external world. So you can't underestimate or you can't understate the importance of understanding how the brain works and what goes wrong in brain disorders. So think about that, because I think we often underappreciate our brains and we don't think about what our brains are doing for us. But in the end, in my mind, the brain is everything about us. Now, the problem is, and to bring this in the context, what you see here in this slide, or in this panel, is really these extensions. Here you have an axon, for example, just a portion of the axon that's terminating, and you see in small green, these small green, these are called vesicles. They release neurotransmitters. And you recall, these are small molecules, these are small compounds that are released, and they can interact with another, this is the dendrite of another neuron, and they can either excite that neuron, stimulate that neuron, or they can inhibit that neuron. But the general problem of the brain, and also to put this back in the context of the astronomy club, the human brain has almost 100 billion neurons. Each of your brains has almost 100 billion neurons. This is about the same number of stars, this is for the astronomy club, in the Milky Way galaxy. So the complexity of our brain, understanding the human brain, is just actually phenomenal when you think about the complexity of the Milky Way galaxy as well. You can put it in the same context, different scales of course, but understanding the human brain is really, really, really difficult. And that's in part why brain health has lagged behind general health in medicine, because it's such a complex system to understand. And it's not for the faint of heart, because I can tell you in my lab and other labs, neuroscientists, we have ideas constantly about what we should do, and many of those fail. Many of those ideas fail. We may spend a year or 2 years working on a project with it, which seems to be a good idea, and in the end, it fails, and so we basically live on those successes. And fortunately, we've been fairly successful in my lab over the years to keep us going for many, many decades now, which is great. But it's a really, really complicated problem. So in addition to the problem of the number, the scale, how many neurons we have, and you recall the neuron is the basic cell type we have in the brain mostly. This is what we focus on, these 100 or so billion neurons. But each of those neurons make about 10,000 connections with other neurons. And so we really cannot wrap our brains around currently the complexity of the human brain. It's too much for anyone to grasp right now. But just imagine 100 billion neurons in your brain, and many of them are active right now, many are silent right now, each making on average about 10,000 connections with other neurons, releasing neurotransmitters. In some cases they're exciting other neurons, in some cases they're inhibiting other neurons. So the brain, this network of neurons in the brain is just an amazing social network that's communicating constantly. in order to process information from the visual fields, auditory fields, learn information, remember that information, and so forth. And so that is our task, really to understand how that happens and what happens in brain disorders, brain dysfunction. So it's a complex problem, as you gather. So these are the depressing, let me turn to Alzheimer's disease now to focus. These are the depressing facts about Alzheimer's disease that I just want to share with you. First, there is a strong genetic predisposition to Alzheimer's disease. And that's true of all brain disorders. Basically, our genetics, our DNA, our genes, they influence the probability that we might, or that we are susceptible to these disorders. Now, Alzheimer's, there's two classes of Alzheimer's disease. One is called early onset Alzheimer's disease, and that is due to genetic mutations in one of three known genes. And if you have a mutation in one of those three genes, you will get Alzheimer's disease. There's no escaping it, you will. So it is genetically determined. Now that only represents about 1 or 2% of all the Alzheimer's cases that exist in our community across the United States or across the world. But we know that there are three genes that cause Alzheimer's disease, and they're early onset. Usually you'll see your Alzheimer's coming on in your 40s, 50s, sometimes early 60s, which is why it's called early onset. The second class is really sporadic or late onset Alzheimer's disease. And this occurs usually in the 60s, 70s, or 80s. Makes up the vast majority of Alzheimer's cases. But it's not dictated by genetics, where if you have a mutation, you would definitely get Alzheimer's. But your array of genes and your DNA will, depending on what you have, will influence the probability that you will get Alzheimer's disease. And we know of numerous genes now that if you have changes in those genes, they will influence your probability of having Alzheimer's disease. And many of you have Alzheimer's disease in your families, as do I, and we'll come back to that in a minute too. There is a 2x risk, 2 times risk of having Alzheimer's if you have a first-degree relative, your mother, father, brother, sister. You are about a 2-fold risk of having Alzheimer's compared to the general population. Many of you have Alzheimer's family. My father passed away of cancer, but at that point in time he had mild cognitive impairment, because clearly it was leading towards Alzheimer's. And actually, in retrospect, it may have been a good thing that he was taken fairly quickly by cancer. His 2 brothers, my 2 uncles, same side, they both died of end-stage Alzheimer's disease. So certainly my genetics are such that I am at an increased risk for Alzheimer's, as are many of you in this room. There are both protective factors and risk factors for Alzheimer's. Aging is the major risk factor for Alzheimer's, as well as certain genes that I just mentioned. Traumatic brain injury, low social economic class, basically educational level is a risk factor. Social engagement, as indicated on the slide, that seems to be protective. So this comes back to the idea you got to use your brain or lose it. So both social and physical exercise is incredibly important for brain health. And so that's part of the message of point number 2 relative to Alzheimer's. The incidence is about 1 in every 9 individuals, age 65, will have Alzheimer's disease or have Alzheimer's disease. About 1 in every 3 at age 85 will have Alzheimer's disease. And as we age and have a larger aging population, that is going to increase in the United States and all across the world, especially in the United States where we have a baby boom. Women are a little bit, about 2 out of 3 Alzheimer's cases are women, probably due to the fact that women live a few years longer than men. That's probably a large aspect of that discrepancy. Maybe there are other factors as well. And about 500,000 people in Florida have Alzheimer's disease. Next fact is the economic cost of Alzheimer's is more than $230 billion per year in the United States. And that's an incredible economic burden. Doesn't speak, and there's no way to even estimate the family burden, or the burden on the individual, the impact it has on the family. You just can't put a dollar value on it. All you can do is figure out the economic burden. So this is an economic train wreck that's coming down our society right now, and it basically tells me we have to solve this problem. I mean, there's no other option. We really have to solve the problem of Alzheimer's. Otherwise, there's going to be a huge economic burden on society. It's only going to get worse. What is Alzheimer's? Alzheimer's always starts as initial memory loss. So this is the initial thing that one sees in Alzheimer's, so memory loss, difficulty remembering. Now, Alzheimer's is one type of dementia. Dementia is a categorical term. So Alzheimer's means you have a memory loss, and then you have some other problem with some brain function. You may have difficulty seeing objects or discriminating objects. You may have a change in personality, either becoming very quiet or aggressive. Some people become confused. And so Alzheimer's presents in individuals differentially. It can be quite different, but it always is diagnosed initially as a memory loss, and then what happens after that could vary from one individual to another individual, depending on how the disorder spreads in the brain. And we'll come back to what we think about the causal aspects of Alzheimer's in just a moment. Let me get back, because this is always an issue. What is dementia? What is meant by dementia? Dementia is a categorical term. Alzheimer's is one class of dementia, but there's also Parkinson's dementia, and there's ALS dementia. Dementia is really that aspect of memory loss plus some other type of defect in cognition. So think about it in terms of cancer. You know, breast cancer is one type of cancer. Alzheimer's is one type of dementia. So dementia is a broad categorical term, but Alzheimer's is the major form of dementia right now. Variability in individuals, I mentioned this already. It starts with memory loss, but it can proceed to different, how it comes across clinically differently in different individuals. First, there is a strong genetic predisposition to Alzheimer's disease, and that's true of all brain disorders. Basically, our genetics, our DNA, our genes, they influence the probability that we are susceptible to these disorders. Now, Alzheimer's, there's two classes of Alzheimer's disease. One is called early onset Alzheimer's disease, and that is due to genetic mutations in one of 3 known genes. And if you have a mutation in one of those 3 genes, you will get Alzheimer's disease. There's no escaping it, you will. So it is genetically determined. Now, that only represents about 1 or 2% of all the Alzheimer's cases that exist in our community or across the United States or across the world. But we know that there are 3 genes that cause Alzheimer's disease, and they're early onset. Usually you'll see Alzheimer's coming on in your 40s, 50s, sometimes early 60s, which is why it's called early onset. The second class is really sporadic or late onset Alzheimer's disease. And this occurs usually in the 60s, 70s, or 80s. Makes up the vast majority of Alzheimer's cases. But it's not dictated by genetics, where if you have a mutation, you would definitely get Alzheimer's. But your array of genes and your DNA will, depending on what you have, will influence the probability that you will get Alzheimer's disease. And we know of numerous genes now that if you have changes in those genes, they will influence your probability of having Alzheimer's disease. And many of you have Alzheimer's disease in your families, as do I, and we'll come back to that in a minute too. There is a 2x risk, 2 times risk of having Alzheimer's if you have a first-degree relative, your mother, father, brother, sister. You are about a 2-fold risk of having Alzheimer's compared to the general population. Many of you have Alzheimer's family. My father passed away of cancer, but at that point in time he had mild cognitive impairment. Clearly it was leading towards Alzheimer's. And actually, in retrospect, it may have been a good thing that he was taken fairly quickly by cancer. His 2 brothers, my 2 uncles, same side, they both died of end-stage Alzheimer's disease. So certainly my genetics are such that I am at an increased risk for Alzheimer's, as are many of you in this room. There are both protective factors and risk factors for Alzheimer's. Aging is the major risk factor for Alzheimer's, as well as certain genes that I just mentioned. Traumatic brain injury, low social and economic class, basically educational level is a risk factor. Social engagement, as indicated on the slide, that seems to be protective. So this comes back to the idea, use your brain or lose it. So both social and physical exercise is incredibly important for brain health. And so that's part of the message of point number 2 relative to Alzheimer's. The incidence is about 1 in every 9 individuals age 65 will have Alzheimer's disease or have Alzheimer's disease. About 1 in every 3 at age 85 will have Alzheimer's disease. And as we age and they have a larger aging population, that is going to increase in the United States and all across the world, especially in the United States where we have a baby boom. Women are a little bit, about 2 out of 3 Alzheimer's cases are women, probably due to the fact that women live a few years longer than men. That's probably a large aspect of that discrepancy. Maybe there are other factors as well. And about 500,000 people in Florida have Alzheimer's disease. Next fact is the economic cost of Alzheimer's is more than $230 billion per year in the United States. And that's an incredible economic burden. Doesn't speak, and there's no way to even estimate the family burden, or the burden on the individual, the impact it has on the family. You just can't put a dollar value on it. All you can do is figure out the economic burden. So this is an economic train wreck that's coming down our society right now, and it basically tells me we have to solve this problem. I mean, there's no other option. We really have to solve the problem of Alzheimer's, otherwise there's going to be a huge economic burden on society. It's only going to get worse. What is Alzheimer's? Alzheimer's always starts as an initial memory loss. So this is the initial thing that one sees in Alzheimer's, a memory loss, difficulty remembering. Now, Alzheimer's is one type of dementia. Dementia is a categorical term. So Alzheimer's means you have a memory loss, and then you have some other problem with some brain function. You may have difficulty seeing objects or discriminating objects. You may have a change in personality, either becoming very quiet or aggressive. Some people become confused. And so Alzheimer's presents in individuals differentially. It could be quite different, but it always is diagnosed initially as a memory loss, and then what happens after that could vary from one individual to another individual, depending on how the disorder spreads in the brain. And we'll come back to what we think about the causal aspects of Alzheimer's in just a moment. Let me get back, because this is always an issue. What is dementia? What is meant by dementia? Dementia is a categorical term. Alzheimer's is one class of dementia, but there's also Parkinson's dementia, and there's ALS dementia. Dementia is really that aspect of memory loss plus some other type of defect in cognition. So think about it in terms of cancer. You know, breast cancer is one type of cancer. Alzheimer's is one type of dementia. So dementia is a broad categorical term, but Alzheimer's is a major form of dementia right now. Variability in individuals, I mentioned this already. It starts with memory loss, but it can proceed to different, how it comes across clinically differently in different individuals. So this is a cartoon. Now let me start on the right-hand side of this slide first. What you see in the upper part of this panel is a healthy brain taken post-mortem. And you can see basically this is the 3-pound brain, and this has the 100 or so billion neurons in the brain. Very, very small, of course, tiny. This is what happens with Alzheimer's. In the end, neurons die, which is why it's classed as a neurodegenerative disorder. Neurons die. Other types of neurodegenerative disorders, Parkinson's causes neurons to die. Huntington's disease causes neurons to die. Frontal temporal lobe dementia. ALS causes certain types of neurons to die, both in the brain and in the spinal cord. But this is a class of neurodegenerative disorders. The neurons are degenerating, they're dying. Now, what is thought to happen, and to explain the way what we see the disease progress clinically, it starts, if you look at this cartoon on the left-hand side, what we're looking at is the human brain bisected right down the middle, and we're looking at it from the inside of the brain. Alzheimer's is thought to start in this area that has this light blue shading. So there is a part of the brain called the temporal lobe that comes right down across your ear. But on the inside, the medial, the middle part of that, is an important memory center. It's called the medial temporal lobe system or the hippocampus. And Alzheimer's is thought to start there. And we know that this area of the brain is important for encoding new memories from a variety of different experiments with humans and other systems. And so that is thought why that you see memory problems initially, because it starts there. But then as you progress down this slide on the left-hand side, you'll see that that blue can spread. That's thought to be the progression of the disease. And depending on how it spreads to different areas, then you're going to affect different aspects of brain function. So for example, this area back here, this is the very posterior part of the brain, right back here. This is the anterior, posterior. The posterior part is where we process vision, for example. Kind of counterintuitive. We basically perceive information through our retina in the eyes, but then that information is processed at the very back of the brain. So if Alzheimer's spreads back there, you might have visual disturbances. If it moves to areas that are involved in mood, for example, you might have changes in personality or mood. So this is the current thought about how the progression occurs and gives rise to the clinical symptoms that we just mentioned. Brain pathology. So this is old history to a certain extent, but what was observed initially in Alzheimer's disease, and basically we began to understand this in the 1980s, is that when you have an individual with Alzheimer's disease, again an individual who initially presents with memory problems, and then that changes to dementia, having other problems with cognition, cognitive aspects, and one can assess these individuals by memory tests that one can do in the clinic and other tests. Initially what was done is to take the brains out of these individuals post-mortem, take those brains, section them, take little bits of tissue, section those, stain them with certain chemicals that allow you to highlight different things in the brain. And what was observed are these plaques, these amyloid plaques. That was one of the first things that was observed in Alzheimer's. What you see here is a brain section, and each of these small brown circles are different neurons in the brain. But outside those neurons, you see these accumulations of this very dark brown, ugly substance. This is called amyloid plaque. And so the initial thought was, in the 1980s, is what is in this plaque? And in the end, it turns out to be an aggregation of proteins that will come back, and one major protein. But this is one of the major diagnostic features, and indeed the only diagnostic, the ultimate diagnostic feature for Alzheimer's, post-mortem examination of the brain. The other thing that was observed were called neurofibrillary tangles. So this is a different stain, and you can't see it, I'm sure, probably because of the lighting, but nevertheless, this is a neuron and stained in a different way, but if you were able to see this under different lighting conditions, there are striations. There are proteins that aggregate into tangles, which you don't see in normal neurons. So these were the two hallmarks of Alzheimer's initially when you looked histologically or basically at the brain. Amyloid plaque forming outside the neurons, in the space between neurons, and within the neurons, this neurofibrillary tangles. And so the task of scientists at the time was to understand what made up these two abnormal aggregations of proteins, the amyloid plaque and the neurofibrillary tangles. And indeed what happened was there was a focus more on the amyloid plaque, and what was found is there was a small protein called A-beta. I'll call it A-beta 42 or A-beta. It's a small protein that is the major protein in amyloid plaque, and that became the idea, that was the major culprit for what was causing Alzheimer's disease, was you have these plaques outside of neurons, and this accumulation, basically dumps of protein, aggregations of protein outside, and somehow they were affecting the ability of neurons to work, to communicate with one another. Now what is A-beta? Where does it come from? It turns out, this is a cartoon as well, and this shows a cell, the neuron, the cell interior. This is the membrane of the cell, which is made up mostly of lipids. And what was discovered over years was there's a protein called APP, amyloid precursor protein. By the way, I forgot to preface this by, if you have questions, this is the task I have right now is to try to present this to you and what we're doing at a level that's somewhere between the Science and Technology Club members, which presumably they're engaged in this, and the local community, which is very interested in Alzheimer's disease. And so if you have questions that you just don't understand, please just raise your hand and we'll try to clarify them right away. But yeah, I forgot to mention that, so please do so. We'll have a conversation. So all of us have this APP molecule on all of our neurons, amyloid precursor protein. And it sits there, and right now we don't really have a complete understanding of what it does. But what we do know is in normal individuals, what happens is this protein is cleaved, is cut up by proteases. These are enzymes that basically cleave other proteins. And this is part of the normal reaction. But in Alzheimer's disease, one of these proteases, it makes a mistake. And it makes a small peptide, or a small protein, in green, shown here, called beta amyloid. It's made a little bit longer than it should be in normal individuals. And that particular protein, beta amyloid or A-beta, has a propensity, a tendency to accumulate. It's sticky. So it sticks to one another and forms these amyloid plaques outside the And as you see here is a cartoon of beta amyloid plaque. And then at the end, if you look in the brain, you see these plaques, the amyloid plaques, histologically. So that became the dominant idea as far as what was causing. It was really the miscleaving of a problem with the processing, we call processing of APP, amyloid precursor protein, and you get this small toxic protein made in Alzheimer's. Why, at this point in case? Probably both genetics and environment playing a role. You get accumulation, and that disrupts the ability of neurons to work and communicate with one another in an essential and normal way. Now, that has led Big Pharma, and I've only shown you one slide, to develop agents that are anti-A-beta. And so one class of agent, for example, they said, well, let's find inhibitors of those enzymes that cleave abnormally, and maybe that will work. And it turns out those don't work because those enzymes have other roles, and if you have inhibitors of those enzymes that are working on APP, you get some side effects that are unwanted. So that didn't work. The more recent large effort that began probably in 2005, 2006, in that kind of range, was to develop antibodies. This is called immunotherapy. Developing antibodies that basically detect A-beta and the amyloid plaque. And what you see on this slide is just a listing. There's no way you can read this. It's a listing of different kinds of antibodies that have been made by Big Pharma, the Eli Lillys, the Mercks, and the Pfizers, and the Biogens, and so forth in the world. And the idea was, well, let's make these antibodies. We can fuse these antibodies into individuals that might have Alzheimer's. They'll basically make it to the brain. Those antibodies will attack the A-beta or the amyloid plaque, just like antibodies do to bacteria. They'll clear it, and Alzheimer's will go away. The bottom line is, these have all failed. And what you'll see is boxed in red, which you can't read, is basically it said, failed to meet primary endpoint, failed to meet primary endpoint, failed to meet primary endpoint. Now, in many cases, if we look at the brain and say, have these antibodies cleared the amyloid plaque? And indeed, they seem to have partially or completely cleared the amyloid plaque from individuals that are undergoing clinical trials. But unfortunately, they did not relieve the symptoms, the memory loss and other cognitive problems that are associated with Alzheimer's. So why is that? Well, there are a number of reasons we could talk about. One, we'll come back to what my lab is doing currently. But one possibility is, for example, many of these clinical trials started with individuals that already have Alzheimer's disease. They had mild Alzheimer's disease. And so one thought in the field is, well, we need to go back, because it starts 20 years earlier, this accumulation of A-beta, 10 to 20 years, decades earlier. We need to go back and start this earlier in time course as a protective therapy, not as something that would actually reverse the disease once one has it. So that's ongoing. But the failures, many of which have cost billions of dollars to Big Pharma, has led to one major unfortunate consequence. One unfortunate consequence, Big Pharma has largely moved away from developing drugs for Alzheimer's disease. Because that's not in their business plan, they've lost money. And they're turning their attention to things that are easier, like diabetes and cancer, very important. But nevertheless, that's a situation where they can make some money, and they have failed big time in developing brain disorders. And so that's a major unfortunate consequence of these failed trials. Yes?
A company called Enzyme, a company
There's the question with regards to a small Italian company. There's lots of small biotech companies right now that are entering the field, and I can't tell you the specifics on that particular company. Some have potential therapies, some not. It depends on the strategy, but I think, as we'll come back to, it depends on your strategy. If we think that A-beta working outside the cell, outside the neuron, is going to be the effective thing, my sense is it's not. Many people think it's not going to be, and we'll come back to why that is. But there's a lot of smaller companies that have something that could be promising. In the end, they have to go through clinical trials, which is very costly and very time-consuming in order to meet FDA requirements for a drug. So that's an unfortunate consequence, but basically the idea of the need to develop therapies is really followed back on biotech companies, smaller biotech companies, not the major companies, and also academics like myself, because we have to solve this problem. And also given rise in the field to the issue of, is this A-beta hypothesis? The fact that this toxic protein sits outside the cell, each neuron is toxic and it accumulates, is this correct? And many in the field thinks maybe it's not the correct hypothesis, and we've been chasing the wrong idea for more than 20 years. And indeed, there's some evidence that already exists that maybe that idea is wrong. For example, one of the important developments in Alzheimer's is there is now a way of imaging A-beta, the plaques in the brain, before postmortem. In the past, you had to basically dissect the brain and basically take it and look for plaques. Now there's a way of injecting a dye that binds to the amyloid plaque, and you use PET scanning, positron emission tomography, in order to see the plaque. And what you see here is indeed Alzheimer's patients, and the red means there's more plaque in this particular image, this PET scan. A normal individual that's amyloid negative is shown here, but we knew before this was done, we knew this before, approximately 30% of the individuals in our age group, from 60 to 90 or more, about 30% of us have lots of amyloid plaque in our brains, but we have no cognitive problems. So it's not a binary decision. Amyloid plaque, yes, you get it. Amyloid plaque, no, you don't get it. It's really a probability. So it's not perfectly diagnostic of Alzheimer's either, the fact to have amyloid plaque outside of the neurons in the brain. So, yes?
Can I just ask a general question? And that is talking about Alzheimer's, but are you also talking about brain degeneration, cognitive degeneration, like frontal lobe degeneration? Or are you specifically talking about only Alzheimer's?
Yeah, the question dealt with, since I'm sure you can't hear, am I only speaking about Alzheimer's right now, or I'm also being inclusive of other neurodegenerative disorders that include ALS or frontal temporal lobe dementia and so forth? I'm specific to Alzheimer's right now, because the amyloid plaque is really the diagnostic, is really what happens in Alzheimer's. In other disorders, for example, Parkinson's, you have a different protein that aggregates called alpha-synuclein and so forth. So they're a little bit, they're all related in one way, but they're different. So I'm speaking only of Alzheimer's right now. So that leads us to a different idea. And this is the idea that we're working on in my lab, and I think we have like 5 or 6 more minutes I'd like to present this idea to you. We're not close to the end in terms of developing therapeutics, but we made some promising inroads. Based upon what I've told you, the idea that this toxic protein, A-beta, accumulating outside of neurons and causing neurons to be dysfunctional may be incorrect. What turns out that another thing besides the tangles that I mentioned, another thing that happens in Alzheimer's disease, and besides the tangles and amyloid plaque, and by the way, what I am telling you right now is and I'll mention this, is consistent across all neurodegenerative disorders, is that in Alzheimer's, that A-beta, it's inside of neurons as well. And it's now known to find its way to mitochondria. What are mitochondria? So what you see here in this slide is in the upper left-hand panel is, again, you see a neuron. And we have ways to paint the mitochondria in green. So you're not seeing all the continuous axons and dendrites, but what you see is the cell body here in green, And then all these are axons and dendrites emanating from that cell body to communicate with other neurons as we grow this neuron in culture. This is a mouse neuron. And we have ways of labeling in a living neuron the mitochondria in green. So we could see the mitochondria. Now, there's other evidence that has shown that this A-beta protein, besides being outside of neurons, it also gets inside of neurons. And indeed, it finds its way inside of neurons, finds its way back inside the neuron. It makes its way to mitochondria. And what do mitochondria do? Mitochondria do lots of really, really important things for all of our cells to function properly, including neurons. But one of the major things it does is it makes ATP, which is the source of energy for all cells to work. Adenosine triphosphate, this is the source of energy. All cells make it, and this is what they use in order to power everything they need. Now it turns out neurons are very energy demanding. Every time the synapses work, they basically require zillions of ATP molecules. And so you can imagine a very, very simple idea, which to me is simple, but no one's ever tested it rigorously, and that's what we are doing currently, is maybe this toxic protein makes its way to mitochondria, causes mitochondria to become defective. Basically, the neuron can't make the ATP that it needs to function. And of course, in the absence of that energy, lack of energy, the neurons are going to be compromised initially in terms of function, and eventually they will die. Now that explains a number of things. That explains, for example, while the immunotherapy, where those antibodies make it outside the cells and they can remove the amyloid plaque, they don't get inside the neurons. So they're not going to remove A-beta inside neurons. So it explains all the failures in the clinical trials. It also resonates with me that mitochondria could be the really critical thing that we need to focus on in order to keep all our neurons healthy, in order to make the necessary ATP and energy for all of our cells to work. So for Matt, just think if you had a compound, a drug that you could wake up every morning, even in a normal situation, and basically you make the mitochondria work 5% better, 6% better, a little bit better. You know, that could be invigorating. Basically like putting SDP for the old mechanics in the audience, SDP in your engine, make it work a little bit better. So that's the general idea. So maybe we have been looking in the wrong place. Maybe we've been looking outside neurons for a long time at these amyloid plaque and toxic A-beta. Maybe we need to think about what's happening to mitochondria. And I want to spend a little more time on mitochondria because they are absolutely fascinating. These small particles, we call them organelles, that basically sit inside of your neurons, all cells, and they provide the energy and other functions for each of your cells to work. Indeed, each of us have, in each of our neurons, we have probably around 10,000 mitochondria. And the amazing thing, which you will not be able to see here, unfortunately, is here we have kind of a real-time video loop of a neuron growing in culture. Here's the cell body and all the axons and dendrites, and the mitochondria are labeled in green. And if you watch closely, they move. They are moved from one place to another, and some zip across the screen very, very quickly. They know where they're going. Some not so much. In a higher power version or a subsection of this shows some of the dynamics that mitochondria undergo. So they're making ATP and doing things good that are beneficial to neuron, but they also fuse together and then they divide. And it turns out that this neurons sense where they need energy. So for example, where they need ATP to work. So you can think of these like local and mobile power generators. Basically, it's like having a generator on wheels and moving it around in the cell or around your gated community, wherever you need. You know, we have a power outage and you have these local mobile generators of electricity that are moving around and providing energy where it's needed for the cell. This is what mitochondria do. So if you have a problem, A-beta is toxic in mitochondria. It causes mitochondria to become defective, especially at synapses. Synapses are not going to work, neurons are not going to work, and again, neurons die. So our task has been, over the last few years, to develop new drugs that really tweak mitochondrial function. This is the STP for mitochondria. Can we make mitochondria work better? And will that offer therapeutic value to Alzheimer's disease and other brain disorders? And indeed, maybe I missed a slide somewhere along the way. Oh, I did. This is a slide, sorry, somehow I got it. of Alzheimer's showing defective mitochondria. It's a cartoon, but nevertheless, this shows the neuron basically with the dendrites and the axons and the structure of mitochondria, which is very, very complex, enlarged. And again, there are about 10,000 or so in each of your neurons. In Alzheimer's, what happens is there's pathology, and if you look in the electron microscope, we could look at mitochondria, and they have normal structure. They're called cristae and other things. In Alzheimer's disease, they become defective. They become disorganized. And indeed, initially they swell, and then they become smaller in Alzheimer's disease. So you get increased fission, increased division, so you get smaller mitochondria in the Alzheimer's brain. So we, at Scripps, Florida, we have an amazing infrastructure for drug discovery that basically embodies or utilizes some liquid handling robotics. We use a lot of robots in order to screen large numbers of random compounds, random drugs, to say, can we find one that works in an assay that we have developed? Yes, there's a question.
What happens to the ATP after they go on fire? Is it consumed or recycled?
Yeah, what happens to the ATP after it's utilized as an energy source? Indeed, ATP is adenosine triphosphate. So for the science and technology nerds, the phosphate has 3 phosphate groups. And when you break a phosphate bond, there's lots of energy released. So it's made from ATP to ADP, adenosine diphosphate. And then that ADP is recycled back into ATP. Yes?
Has there been any stem cell research?
We have some going on, so I'll just touch on that at the very, very end. Okay? So right now we basically been looking for drugs that, I'll take one more question. Yes?
Do drugs such as Aricept and Namenda do anything?
Yeah, drugs, so that's a general question. Drugs like Aricept and Namenda, these are drugs that don't work on mitochondria. They do other things, and they do have efficacy. The clinical trials for these drugs basically say you could delay the cognitive impairment by 3 months to 6 months, maybe 9 months, which doesn't sound like a lot, but if you're an individual with Alzheimer's or family, certainly that could be beneficial, having a little bit better cognition and memory for a longer period of time. So they do have, clinical trials have shown they do have efficacy. So that's really the initial line of defense right now, are these drugs. Now they have different mechanisms of action. They work differently than what I'm talking about, but they do seem to delay, they don't stop it, the progression of Alzheimer's disease. So these are just some photos basically showing that we do liquid handling robotics, and we do lots of assays and say, can we find new drugs that basically tweak and potentiate mitochondrial function? Sometimes we use a very large robot at Scripps, and this allows us to screen through randomly hundreds of thousands of molecules. Sometimes, depending on what we do, we could only screen through thousands of molecules, and indeed, our level of screening right now is through thousands of new drugs to say which ones might have effects. And indeed, we now have drugs that we've isolated that, and this shows what's called a dose-response curve. It's kind of, you could ignore that, but we have drugs, for example, that we could put on neurons in culture, and indeed, they increase the number of mitochondria by about 10%. That could be therapeutic. So if Alzheimer's, if you're losing mitochondria, they're increased fission, and they become defective during that period, if you have a drug that potentiates, makes more mitochondria, could be effective. Could be effective. We also have, when neurons go bad, the mitochondria become fragmented. You can see these round circles. And we have drugs that actually restore the normal shape of mitochondria. They basically keep them from becoming defective. So we have isolated about anywhere from 7 to 12 drugs that we're really focusing on right now to say, can these have efficacy in Alzheimer's disease? Initially in model systems. And indeed, the first level is really cells in culture. And what this shows is just a bar plot. And what we've done here is we've taken neurons in culture, grown them, we've put onto those neurons this toxic molecule, A-beta, to try to mimic as much as possible what happens in Alzheimer's disease, this toxic A-beta molecule. And indeed, this is the size, this is a measure of fragmentation, mitochondrial fragmentation. This is the normal range shown here in the black. And indeed, when we put on the toxic protein A-beta, this is only for a 24-hour window. if in human life you have it for 20 years, much different. So the potency may not look high, but this is significantly different. There's more mitochondrial fractionation. And indeed, if we put on the toxic protein, and here we have 5 different drugs, we've simultaneously added the drug and the toxic protein A-beta, and basically it keeps the mitochondria from becoming fragmented. So this is the first line of test. Are these drugs working in a cell culture system? And indeed, they seem to work. Now that's not the final test. It was mentioned, are we working on stem cells? So it's now possible to take human fibroblasts, for example, skin punches, or other types of cells from an individual with Alzheimer's disease or other disease, or normal individuals. Take those cells, even let's say fibroblasts, even skin cells, and we do what's called de-differentiation. You take those cells which are now specific to become fibroblasts, and you de-differentiate them, which means you take them back to a primordial state, pluripotent stem cells, which means they are no longer fibroblasts, but they have the ability to become any kind of cell, or many different kinds of cells. And there are ways in the lab now that have been developed over the last 15 years or so to cause those cells to now differentiate into neurons. And that's indeed what you see here. They don't come in multicolor as you see here, because this multicolor is stained with antibodies that allow us to detect neurons. But these are human cells, neurons grown in culture. They were de-differentiated, so they became pluripotent, or basically able to make many cells, and then we caused them to differentiate back into neurons. And indeed, our drugs are now being tested in these stem cells in culture. Now that also, we don't know the answer to that yet, but that's where we are. What's happening in this forthcoming year as well, because this is a long process, you have to go through multiple steps, is we have both Drosophila, these fruit flies. We have engineered flies, believe it or not, and we can test their learning and memory capability. We can look at their mitochondria. We can cause expression of this toxic protein, and we have Drosophila models for Alzheimer's disease. And neurons die in these models as well. And so it gives us a laboratory model before even thinking about human trials to test these drugs. The nice thing about fruit flies is they're compared to mice or rats, they're very inexpensive to work with, and they're very fast to work with. It takes 6 months to get tested in a mouse. Flies, we get it in 2 weeks. So you go through more rapidly through a fruit fly and say, are these drugs having efficacy? Are they working in a fruit fly model for Alzheimer's disease? And then progresses so with those that are into a mouse, which takes much longer and is more costly as well, because we have to keep those animals for 6 to 9 months in order to do behavioral analysis. But we have mouse models for Alzheimer's as well. So we currently have these drugs. They seem to be promising at the level of working in cultured cells in keeping the toxicity of A-beta away. We know in flies too, it seems to be keeping the toxicity from mitochondria away. But this is a progression. It's not a short-term task, and we'll be doing this probably for the next 5 years as we tweak these molecules and make them more effective as we go through this progression, and hopefully, eventually, into clinical trials for humans. So this is my group. Just wanted to thank you for your attention. We can entertain more questions at this point in time. For those of you who are interested, I have a millennial. I have many millennials in my lab that know lots about Facebook, and I don't know anything about Facebook, but there's one millennial in my lab that keeps track of. We have a Facebook page now, which is fairly new, and try to keep the public up to date with what we're doing. So I'm told to tell everybody, please visit us and like us on Facebook. You can listen to that, or you don't have to. I'm still agnostic about whether this is beneficial or not, but it's the Ron Davis Laboratory at Scripps Research. And you can see this is the group, about 16 people in the lab, equally divided, about half working on therapeutics and half on basic science, learning and memory. So thank you very much. Yes.
Question, does a brain aneurysm come under the umbrella of Alzheimer's?
No, brain aneurysm is really a vascular problem, so it doesn't come under the issue of the definition of Alzheimer's disease. But you have brain aneurysms and you have vascular problems. So you know changes in how the vessels work. You have basically if they leak, or if you get one that basically leaks big time, you can have vascular dementia as well. That's one form of dementia I didn't didn't mention. It's problems with the blood vessels in the brain and basically causing problems with brain function.
Dr. Davis, you've got a lot of awards over your lifetime. This one is a lot of appreciation.